Hello and welcome to all. I'm Dr. Charlie Roberts, Director of the Comprehensive Cancer Center here at St. Jude Children's Research Hospital. And with my co-host and actually our speaker for today, Deputy Director Dr. Charles Mulligan, I'm delighted to welcome you to the fall 2021 season of the Science of Childhood Cancer Lecture Series. So this week actually marks the one-year anniversary for the third season kickoff of this series. And it's been truly fantastic to see the enthusiastic engagement that we've received from both the national and global scientific research community over the past year. In fact, since the series began, we've had more than 3,000 individuals join lectures from all over the world. And in just the last 3 weeks since announcing our new speaker lineup, we've had an additional 500 people register. So I want to say welcome to all, and we're looking forward to another great season. For those of you who are new to the series, before we begin, I'd like to just take a few moments to introduce you to St. Jude Children's Research Hospital. Next slide. This is our campus in Memphis, Tennessee, which spans 90 acres and has more than 5,000 employees. While St. Jude is well known as a children's cancer hospital, not all are as familiar with the scope of our scientific research enterprise. Next slide. Science is at the heart of our mission to advance cures for pediatric cancer and other catastrophic childhood diseases. We have a unique funding model that offers unparalleled investments across the spectrum from basic, translational, clinical, and all the way to population science. And indeed, with the launch last month of our new strategic plan, we will be investing $11.5 billion over the next 6 years in pursuit of our mission. Next slide. With a faculty of 300 working in disciplines that range from computational and chemical biology to cell and molecular biology, immunology, oncology, epidemiology, we span a wide spectrum of science, just to name a few. Next slide. In fact, we're growing. We have recently opened our new Advanced Research Center, which is enabling the largest faculty recruitment effort in our history to expand our cancer-focused basic and translational research faculty at St. Jude. Next slide. As well as the cutting-edge science conducted on our campus, to accomplish our mission, we're committed to sharing data and working collaboratively with scientists everywhere to advance discovery and treatments for children around the globe. Thus, we're delighted to have you join us over the next 15 weeks as we bring you lectures from some of the world's leading experts in the biology, mechanisms, and treatment of pediatric cancer, as well as pediatric cancer survivorship. We're also pleased to announce a new component of the series this season, 2 joint Rising Stars of Cancer Research lectures that will feature thematically related talks by early to mid-career investigators who are conducting innovative work in their field. So stay tuned for those 2 sessions on October 7th and November 11th. Next slide. As a brief housekeeping note before we begin, if you have a question at any point during the talk, you don't need to wait until the question time comes. Just at any time, please enter it into the WebEx chat box using this icon and send it to all panelists. If any technical issues arise, you can contact SOCC support email address that's shown here. Next slide. And so now it is my great pleasure and honor to introduce Dr. Charles Mulligan, who I know quite well as he is the Deputy Director of the St. Jude Comprehensive Cancer Center, as well as co-leader of our Hematologic Malignancies program. Dr. Mulligan's research examines the genetic basis of acute leukemia, particularly acute lymphoblastic leukemia, with the goal of identifying the constellations of mutations that drive leukemogenesis, influence treatment response, and to use this information to develop novel diagnostic and therapeutic approaches. He has defined the landscape of genomic alterations that drive the pathogenesis of ALL, developed multiple experimental models of leukemia that faithfully recapitulate the genetics of human ALL, and has identified multiple new treatment approaches that have been implemented in clinical trials. These studies have led to numerous landmark publications that have truly profoundly influenced our understanding of the genetic basis of this disease. Indeed, he's an author on 237 primary research publications thus far, many in the very highest impact journals. I'll just briefly mention a few of his honors. He's been elected to ASCI, the American Association of Physicians, Australian Academy of Health and Medical Sciences. He's the inaugural recipient of the St. Baldrick's Foundation Robert Arcisi Innovation Award. He's received from the American Society of Hematology the William Damashek Prize that recognizes young investigators who have made major contributions to the field. He is extremely well funded. He has an NCI R35 Outstanding Investigator Award, a U54 grant, multi-PI grant from NCI. And I don't know how he finds the time, but he's also the PI of a training grant in hematologic malignancies. He's got awards from LLS, Alex's Lemonade Stand Innovation Award, and a $5 million Crazy 8 Award. He's got indeed 4 pages of completed grants on his CV. On a personal note, I've worked closely with Dr. Mulligan for the last couple of years as he has served as Deputy Director of our Cancer Center. He possesses a remarkably thoughtful intellect, is a master at handling myriad tasks and roles, and delivers top quality of work. And on top of that, he's a wonderful person with a keen sense of humor. And it's my true honor to turn the
Charlie, thank you so much for that really kind introduction. Just please let me know that you can see my slides and hear me okay.
Yeah, we can see them.
Fantastic. So it's a real honor to give the first talk in this series. It's been such a spectacular event so far. And this has really been such an odyssey of discovery over the last decade or so in understanding what is the commonest childhood malignancy. I can't cover everything I'd like to in the time I have. So today I've taken a somewhat selective approach. I apologize for my personal biases in advance, but I wanted to touch on some themes that I think are important and exciting about how genomics have really revolutionized our understanding of the genetic basis of this disease, how it's clinically really important, both the risk stratification across the age spectrum, as well as giving us insights into potential new therapies, and then to really do a deep dive on an area that's been of interest for a long time for me and St. Jude, and that is this really problematic group of leukemias that defy classification, and that'll be the latter part of the talk. So with that, by brief introduction, I think most people on the call, the talk today would know that ALL is a great success story of cancer therapy, but there's a paradox in that despite the overall favourable cure rates that exceed 90% in children, it remains a leading cause of death in the young from cancer. When relapse occurs, it's problematic, and this problem increases with increasing age, where the outcomes for ALL are poorer through adolescence and adulthood. And indeed, a lot of what you'll show you today is not just from studies in children, but right across the age spectrum, which has been a very powerful approach. And of course, we have a clinical imperative that even though we cure most children, this comes at a cost of toxicity, and this is because our therapies have relied on intensification of non-targeted agents, not targeted to mutations or the pathways they deregulate, with rare exception, that being the kinase inhibitors and now immunotherapy in some subsets of leukemia. So let's go back 2 decades really to when, about the time I started working in the field, where our classification of ALL, of B-ALL and also T-ALL was really driven by conventional cytogenetics and targeted molecular approaches, which identified key subtypes in about 2/3 of cases. But a substantial minority of cases did not have one of these sentinel cytogenetic gross changes, such as aneuploidy or a translocation. And we also knew that there was often a very long latency between initiation of disease, which we know for many subtypes can occur prior to birth, to clinical disease, which can manifest several years or indeed decades later. And also the initial attempts to model what we believe to be initiating lesions in mouse models were met with limited success, with low penetrance, suggesting that there must be additional texture in the genome of leukemia that contributes to the development of the disease. Now, to my mind, the revolution in genomics really began with this landmark study from my previous mentor and the CEO of St. Jude, Jim Downing, almost 2 decades ago now, which was really eye-opening. So this was using microarray gene expression profiling, which was deployed at the time to many different diseases. And it had several key implications. The first is that if we do take the known subtypes of disease, they're associated with very distinct gene expression profiles or transcriptional outputs. And this holds true today, as I'll come to in a moment. But there was additional lessons to be learned here. There were clearly new subtypes that defied identification until next generation sequencing became available. And these studies started to identify the notion of phenocopies, that is, cases that had gene expression profiles that mirrored canonical subtypes, in this case here, Philadelphia chromosome positive leukemia, with many cases that had a similar gene expression profile. And of course, we now know that's PH-like ALL. Now, if we come to the present, here is an unpublished study under review at the moment where, in collaboration with many groups and individuals, particularly with computational biology, Sam Brady and Jingwei Zhang and Catherine Roberts from my group, we've studied over 2,700 children, adolescents and young adults with some level of genome-wide sequencing data. And this reinforces what was previously known in part, that the mutational burden of ALL is very low, one of the lowest burdens of any tumours in children or adults, with on average 4 non-silent alterations per case. And if you look at the texture below here, one can see that the majority of these cases are not simply sequence variants, but most subtypes have leukemogenic rearrangements or fusion oncoproteins, and often the number of structural changes, copy number changes such as deletions, outnumbers the number of sequence alterations. So this is a very important lesson because it suggests that to understand the genome, one has to look broadly beyond sequence mutational profiling. One can also see here, I'll come back to this later, that if we look at different subtypes, they have different textures of mutational signatures, which is quite interesting, and we'll come back to that a little later. So there are various ways to think about it, at least in my mind, about the genomic drivers of leukemia. That is, what are the drivers that define subtypes? I'll come to that in a moment. What are the pathways? But also, what's the sequence of evolution? And a number of studies performed now over really 20 years have begun to stitch together, if you like, these genetic alterations to understand the sequence of genetic alterations that we believe initiate the disease and then result in a fully fledged leukemia. It's a disease of 2 genomes, the inherited genome and the somatic genome, and we increasingly appreciate that perhaps the majority of children, at least, have one or more inherited variants, either non-silent SNPs or, less commonly, mutations that have a subtle or more dramatic effect in predisposing to the disease. Then there's a sequence of events, so often translocations, but also aneuploidy that start the process going. There is typically an arrest in lymphoid maturation, both in B and T lineage disease, and then events that disrupt a number of key pathways, tumor suppression, signaling, chromatin regulation, and so forth. And then finally, leukemia develops, and we now know that in most individuals that there's not one clone but many. And typically, these leukemic cells in individuals share a number of truncal or clonal alterations, often that are deregulating transcription factor genes, but also other pathways as well. And when disease does evolve and is not cured, there is collapse of this clonality, typically with emergence of one clone, and often with acquisition of additional mutations along the road that are enriched for from diagnosis or are picked up along the way. And we now know that some of these can actually be promoted by the administration of the therapies that can be otherwise curative. I'm not going to speak much about the biology of relapse today, but clearly an important area. So the first area I want to touch on is classification of the disease, because it has such profound implications for how we think about biology, for how we classify the disease, and how we think about novel therapeutic approaches. Now, although arguably whole genome sequencing is the single most comprehensive approach, it's really transcriptome sequencing that's provided the greatest revolution, if you like, in the classification of disease. And over a space of about 5 years, there were a number of studies of cohorts, often not that large, but they pulled together childhood cohorts, sometimes adult cohorts, often cases that were not classified, and they started to identify a number of rearrangements, often of transcription factors, that appeared to classify new subtypes of disease. In the last couple of years, we and others, there have been several of these studies, have been able to move a little beyond this and not look at selected cohorts, but very large cohorts to try and get a more complete understanding of the subtypes and their drivers. I touched on one of these studies before when I was showing the mutational burden plot, and that's intersecting with this study here, which is looking at the subset of cases, now including adults, that had transcriptome profiling. So this is about 2,000 cases, 2/3 childhood, 1/3 adult. This looks like a single cell t-SNE plot. It is a t-SNE plot, but here we're looking at bulk transcriptomic data where each individual data point is an individual case, and they're color-coded by subtype. And one can see the known subtypes that were well established, both aneuploid and many of the rearrangements, but a number of new subtypes that were not appreciated before by conventional approaches. As I mentioned, several of these are defined by rearrangements targeting transcription factors, many of which have been inappropriately expressed now or hijacked in the lymphoid lineage in a driving disease. The scale of this study enabled some newer, smaller subtypes, often of prognostic relevance, to be identified. But also this study allowed several new lessons to be learned. One of which is transcription factors that drive lymphoid development normally we know are altered in ALL, but now we're seeing that specific alterations can define distinct subtypes of disease, and I'll come back to this. We see additional alterations, point mutations in specific transcription factors that define other subtypes of disease very crisply, at least by gene expression, although we still have a limited understanding as to why these specific sequence mutations that are no less or more deleterious than others are defining distinct subtypes of leukemia. And then we have this large group of cases that we call kinase-driven or pH-positive and pH-like, which perhaps are one of the more clinically important and also genetically diverse subtypes of leukemia. Now, I can't cover everything today, and I want to touch on other themes as well, but I want to make some points as to why these observations have been made and why they're important. So the first is to make the point as to why sequencing is important and why these subtypes weren't identified. And that's because many of them simply aren't resolved by cytogenetic analysis. Some of the areas of rearrangement are not poorly resolved by karyotyping. Some of the alterations are polygenic. That is, we move from a single fusion protein defining a subgroup to multiple fusions that converge on a subtype. An example of that in a moment. And as I mentioned before, we appreciate this phenomenon of phenocopying. So here's one example, and we see this repeatedly with many of the new subtypes of leukemia, where one transcription factor not otherwise considered to be important in lymphoid development, here a muscle transcriptional regulator MEF2D, is rearranged to a number of fusion proteins, BCL9, and a number of genes that interestingly are involved in RNA processing and handling, defining the subtype of disease with distinct immunophenotype and distinct gene expression profile. And a reason I wanted to show this is that as we start to profile these subtypes and look at the genomics and some of the biological features in models, we can often identify potential vulnerabilities that perhaps are not what we typically consider to be a targeted approach, but nonetheless are potentially attractive. So for example, here, MEF2D in muscle development activates the expression of HDAC9 and forms a reciprocal feedback loop with downregulation of MEF2C. And indeed, of course, there are inhibitors of HDACs, and if we take primary leukemic cells from these patients here in xenograft models, we see potent activity of histone deacetylase inhibitors such as panobinostat. So not a selective approach by any means, of course, these are widely used agents, but markedly more active in this form of leukemia than many other subtypes of BALL. An important point is this notion of phenocopying, and of course we now appreciate this for pH-like leukemia, but we see it for other subtypes, indeed almost all subtypes of leukemia. And one of the newer forms of this that was recently described is ETV6-RUNX1-like leukemia. ETV6-RUNX1 leukemia being, of course, one of the most common forms of childhood leukemia, defined by this single fusion oncoprotein. And shown here in grey are the cases that are, as you can see, clustering by gene expression, but don't have this fusion protein. And most subtypes show this to greater or lesser predominance. Of course, the standout being pH-like leukemia, where the likes outnumber the cases with a canonical fusion, at least in children. So what does this mean when we look at the genomics? Well, often the fusions make sense, although there are new findings as well. So there are various reasons why these leukemias are phenocopying ETV6-RUNX1 leukemia, but often it's because they have fusion oncoproteins that involve ETV6 itself, or other ETS family proteins or other transcriptional regulators that presumably have the same transcriptional output. Now, a key thing that I think is quite new in the field of ALL, at least, is the notion that single point mutations can define subtypes of disease. And as I showed you before, we've seen at least two examples of this. There are indeed more, but two standouts are in two key transcription factors that regulate B cell development, that is PAX5, or lymphoid development writ large, that is ICROS or IKZF1. And we see these two specific point mutations defining these subtypes of disease. Now, this is making an old story new again. You know, PAX5 is in fact the first focal target of genetic alteration that we identified well over a decade ago now in our first genome-wide study using SNP arrays, where we saw a remarkable diversity of deletions of the gene, of point mutations and fusion proteins. But with the exception of the fusions, we kind of considered that these were largely secondary events that were disseminated across the landscape of BALL, but weren't actually initiating drivers. Now we know that's not true. So this is here first showing you a protein lollipop plot, if you like, showing the PAX5 mutations that are in this group of leukemia, the P80R group, where we see these cases have this hotspot of mutation in the DNA binding domain. And then if we look at the landscape of other mutations in other cases, again, it makes this point that's still not well understood, that there are often other point mutations that at least in vitro appear to be just as deleterious, but they don't define a subtype of leukemia. What we do know, however, is that in this subtype, it's typically biallelic. Firstly, if we look at the human data, we see that through deletion or LOH, or less commonly a loss of function mutation, the activity of the second or wild type copy of PAX5 is lost, leaving hemizygosity for this P80R mutation. So this for us was interesting. We wanted to formally test then this notion that a single point mutation can be leukemia initiating. And to do so, we initiated this some years ago when we were just starting to use genome editing to make knock-in mouse models. And we chose two PAX5 mutations, the first of which is the P80R mutation that I've mentioned, the second of which is a mutation that's seen in rare familial leukemia. And what we saw was quite striking, and at least striking for us that work in BALL, where there are typically very few penetrant models, where we saw that mice heterozygous for the P80R mutation developed leukemia with approximately 6 months latency, and those that were homozygous with a substantial reduction in time to onset of leukemia. The leukemias would be lymphoid, and then if we close the circle by performing genomic analysis of the mouse tumours, we see very similar findings to what we see in human disease. Here this is showing array CGH of mouse PAX5 showing focal or broad deletions of that second copy in heterozygous mice. Or in a homozygous mouse, we even see amplification of the mutation, providing strong experimental confirmation that firstly, this is leukemia initiating, and secondly, it truly is a stepwise evolution to develop disease. Let's come back to now an unpublished story that is interesting. And as we were thinking about our data that I opened with, looking at the overall genomic landscape of leukemia and mutational burden, we asked the question, can we address the basis of the genesis of hyperdiploid ALL? So this is the second subtype that predominates in children, where we see these stereotyped aneuploidies of multiple autosomes and often the X chromosome, the basis of which is not well understood, both in terms of why you generate the aneuploidy, when it's acquired, how it interacts with the acquisition of sequence mutations and the order in which those two events occur, and the time at which these events may originate. Is it prenatal, postnatal, or which of these predominates? So to do this, firstly, Sam Brady who analysed the data, was looking at the mutational plots as shown here. As I mentioned before, if we look at the aneuploid cases, hyperdiploid, and near haploid ALL, as well as cases with complex changes of chromosome 21, we see that many of these cases have enrichment selectively of this UV mutational signature. And so we thought combining the DNA-wide data, genome-wide DNA data of copy number profiling, the presence of aneuploidy and mutational data, can we look at the sequence of acquisition? Could we perhaps ask which of these two scenarios might be correct? Is it that we see initially or continuously the acquisition of sequence mutations and then there's gain of chromosomes together or separately? Or is there perhaps an early event where there's the generation of aneuploidy that's then followed by sequence mutations? And the answer is that we think there is, in most cases, a big bang, if you like, event where early in life, and we think prior to birth, there's the acquisition in a synchronous fashion of these multiple aneuploidies followed by sequence mutagenesis. So if we look at the UV-induced mutations of these hyperdiploid cases, what this is showing is that these UV-induced mutations are occurring on one chromosome, not two chromosomes or three chromosomes, And that strongly suggests that they're occurring after the triplication of these chromosomes, so after the initial generation of aneuploidy. If we then look at all aneuploid chromosomes, so all of the triplicated chromosomes, and look genome-wide at the sequence variants that are on those chromosomes, we see that the majority of cases look like this, where the variant allele frequency is usually around 1/3. So again, they are occurring on one chromosome, but not 2 or 3, and this accounts for over 2/3 of cases. Less commonly, we see different patterns where there's a late acquisition of aneuploidy or a more complex pattern. So this for us was very interesting, as it provides conceptual insight into the origin of these aneuploidies and the time at which they occur. Now, as I mentioned, I can't cover everything, but I wanted to come back now to make a couple of points about the clinical implication of this novel genomic taxonomy of leukemia. The first is that these genomic drivers vary substantially by age, with disappearance of these good risk prognostic groups with increasing age into adulthood, largely, and an enrichment of kinase-driven subtypes and then additional subtypes such as MLL rearrangement and low hypodiploidy to a much more parsimonious landscape of subtypes in adulthood. And as many of you would know, these subtypes are poor prognosis and associated with a high risk of relapse. So a key point here is that we've been asking this question for some time: how much of the declining prognosis with increasing age is due to biological features in genomics? And the answer is much of it is driven by this. So this is supported by 2 recent studies from children on the left, from adults on the right. So Total Therapy 16, the last completed ALL frontline study at St. Jude, where we see many of the new subtypes are associated with favourable or unfavourable outcome, and similar findings, I think even more striking, in adults, where we had limited understanding of prognostic drivers, where we're now seeing some subtypes that have prognosis and outcomes that are approaching the favourable outcomes of good risk childhood ALL. What does this mean for therapy? I won't go into this in a great deal of detail, but the answer is a lot, both in terms of perhaps reducing therapeutic intensity for some of the recently described good risk subtypes, but also, as you'll see at the bottom, for the majority of subtypes, we have either preclinically validated approaches to target many of these new subtypes or genomic drivers, or approaches that are perhaps mutation agnostic, which have been shown to be highly efficacious in one or more forms of this leukemia. This, of course, poses challenges in terms of clinical trial design, but it does make the point very strongly that this is an exciting time in therapy for ALL, both in children and adults. Now, before closing out this section, I want to return to, again, an old story that's becoming new again, and that is one of the first subtypes I mentioned, and that is PH-like leukemia, which I would argue remains the greatest unmet need in B progenitor ALL. So this was described through complementary approaches by a combined study between St. Jude and the Children's Oncology Group, and also a European consortium led by the Dutch, that basically made the observation that there are cases that cluster with pH positive cases that have a similar gene expression profile, but they're pH negative, hence the term pH-like leukemia. And a lot of work from many groups has remarkably consistently shown that this form of leukemia is high risk, as evidenced by high rates of MRD or poor outcomes. And it's also really common, as shown on the top left here, that with increasing age, pH-like leukemia rises in prevalence to predominate, particularly in the older age spectrum. It is one of the most genomically diverse forms of cancer that's been identified, where at least 16 cell surface and intracellular cytokine receptors and tyrosine kinases are targeted by often rearrangement, but not always rearrangement, sometimes other alterations such as sequence mutation and deletion that activate canonical signaling pathways that, at least at the preclinical level, are targetable with the cognate tyrosine kinase inhibitor. So where do we stand with this? Well, that's a very complex table, lots of partner genes, but another way of showing this is visually to show that these alterations can be distilled into their key pathways, the commonest of which is activation of the Janus kinase STAT signaling pathway, the JAK-STAT pathway, where there are many alterations that have their own genomic nuances that are fascinating, but I won't go into them in detail today. But they typically involve either rearrangements that deregulate CRLF2 or, less commonly, the JAK family of kinases themselves, and also the erythropoietin receptor, often with additional lesions that facilitate signaling, point mutations of Janus kinases and other lesions. The next most common is the ABL signaling pathway, as shown here. And then increasingly, we're seeing at lower frequency but greater diversity these other kinase targets of rearrangement, often with new genomic structures to their deregulation that have not been previously seen in cancer. For example, chimeric fusions with FLT3, FAC rearrangements, and others. And a point to make here is that, yes, we know that when we introduce these lesions experimentally, when we study patient cells, we do see typically activation of the predicted primary signaling output, but often activation of parallel signaling pathways that are also inhibitable. And indeed, we now understand that combined therapy with multiple kinase inhibitors, or perhaps additional therapy with venetoclax, is much more potent in suppressing leukemic cell proliferation. So where do we stand with this? So starting with a very good, a standout example here is ETV6-NTRK3, which of course is rearranged in other cancers, not just leukemia. But this is really a singular example in PH-like leukemia where this truly is a monogenic driver. It induces leukemia in engineered mouse models that we've shown, but I won't show today. It's in this paper, and also in cell line models. And that activity and signaling is completely shut down with the appropriate inhibitor, a single inhibitor like larotrectinib, or a combined agent such as the Plexicon agent shown on the top right. If we treat xenografts, we can essentially completely suppress leukemic cell proliferation. Not shown here is that if we continue therapy for long enough, we eradicate leukemia and it doesn't come back. So a standout example where there's exceedingly strong rationale for adding these drugs in the presence of this fusion. That's now been shown in case reports that continue to emerge, where these agents are showing great activity, often in exceedingly heavily pretreated patients with multiply relapsed leukemia, showing great response to agents such as larotrectinib. And very similar findings have been seen now in larger series. We reported some of these data a decade ago, but now here's another very nice study from a couple of years ago from Europe, showing in a well-studied series incorporating genomics that, again, in a high-risk context of relapsed and refractory leukemia, that ABL class fusions treated with drugs such as dasatinib can show not just responses, but often deep molecular responses that are durable. So very strong rationale to treat with these agents. Here is the bad, and so this is unfortunately the more common form of PH-like leukemia, the JAK-STAT pathway, the majority of cases of which are driven by rearrangement and deregulation through enhancer hijacking events of this gene CRLF2 cytokine receptor-like factor 2, which forms a heterodimer with one of the common interleukin chains, interleukin-7 receptor alpha, and then activates this cascade of signaling, primarily JAK-STAT, but then with bleed through to other pathways, RAS-RAF-MEK-ERK and the PI3K pathway as well, to drive leukemic cell proliferation. Notably very common in Down syndrome, BALL, as well as PH-like leukemia. So the unfortunate data is shown here where now there's great diversity in the JAK-STAT pathway of PH-like leukemia, but typically we show that the more conventional chimeric fusions, two examples of which are shown here, show often pretty good responsiveness to a single agent TKI, particularly if it's combined with a bit of backbone chemotherapy like dexamethasone. But when we examine CRLF2 rearranged xenografts, we see outcomes much more variable if they are responsive at all, and many xenografts do not respond to TKI therapy. So we and others have known for a long time that we need a better approach to try and induce response in this high-risk form of leukemia. Now, why don't these patients respond? And one reason is that the existing type 1 inhibitors that bind the active site of the JAK kinase domain are bypassed because we can see heterodimerization with other JAK family members like JAK1 and JAK2, and we can see transactivation and persistent signaling leading to a persistent output of these signaling pathways and bypass of activity. There was some interest and excitement for allosteric type 2 inhibitors that showed in preclinical models some improvement in activity, but that's not been confirmed. It's certainly not in our hands, and these agents have not progressed to the clinic. Now, recently, as many would know, there's been quite a lot of excitement with the paradigm of targeted protein degradation. One approach of which are these PROTACs, or proteolysis targeting chimeras, the approach of which is to couple a ligandable domain through a small molecule via a linker to another moiety that interacts with an E3 ubiquitin ligase. And this allows for direct interaction between the ligase and the protein of interest, ubiquitination and proteasomal degradation. And so this has several theoretical advantages in terms of, rather than just being kinase inhibition, in this case, it can, through protein degradation, result in a different biochemistry, stoichiometry, and pharmacokinetics, and more deep and prolonged inhibition of JAK signaling. So we've sought to embark on this in the last few years. This is our first foray into targeted protein degradation. This has been a really wonderful collaboration that's been so enjoyable with our Department of Chemical Biology and Therapeutics here, with collaborators Zoran Rankovic and Marcus Fischer in particular, where Marcus solved the crystal structure of the JAK2 kinase domain, coupled with the inhibitors baricitinib and ruxolitinib. And this allowed a couple of observations to be made. Firstly, it showed that the JAK inhibitors adopt a somewhat unexpected conformation when they're bound to JAK2, but importantly, it allowed selection, of an appropriate area to couple the linker to then design the PROTAC. And so this is the general structure for the PROTACs that were embarked upon using cereblon as the E3 ubiquitin ligase of interest. And then this is our initial series of 60 PROTACs, but many more have now been synthesized, with continued structural resolution showing here some of the first PROTACs, with the linkers now extending out into solvent, as shown here. And so some of the data are quite remarkable. So here, this is coupling a drug such as ruxolitinib through a linker to an IMiD such as pomalidomide, and both of the parent molecules essentially are inactive. But if we couple them together in a PROTAC, we see many thousand-fold increased potency in cell lines and primary leukemic cells, here using MHH-CLL4 as a paradigm of serial of 2 rearranged leukemia. So we've extended these studies. We've taken several combinations of design, the details of which I won't go into today, but we've shown that these agents are often highly potent and effective in xenograft models, and notably their activity is much greater typically than the parent compounds, which usually are often inactive, like ruxolitinib, for example, and the type 2 inhibitors. It's increasingly appreciated that these PROTACs often degrade additional targets, such as a component of the translational machinery GSPT1, but in our ongoing studies, we've been able to dissect out and identify degraders of the Janus kinases that in fact spare GSPT1, often with retention of JAK2 inhibition and cell killing. So there, I want to pause for breath and perhaps a sip of water. And for the remainder of the talk, switch to a completely different theme. My title of the talk was Deconvoluting the Origins of Acute Leukemia. And I mentioned that I wanted to spend some time talking about this area that's been of interest for some time. And perhaps you might consider it the forgotten leukemias. We in the field are typically preoccupied with acute lymphoblastic leukemia or acute myeloid leukemia. But these leukemias that defy neat classification have been a problem for a long time. So these are called lineage ambiguous acute leukemias. They're typically diagnosed with non-genomic approaches, immunophenotyping in particular, to various categories that have been basically the best that we can do, but are pretty unsatisfying in terms of understanding biology. For example, early T-cell precursor leukemia, mixed phenotype acute leukemia, and also leukemias that really lack markers of differentiation that are called acute undifferentiated leukemia. We have considered that they may originate from a progenitor because of this, these ambiguous features, but there's not been a lot of data to formally implicate that an early hematopoietic stem cell, or perhaps a somewhat more differentiated progenitor, is truly the cell of origin, and that's really what I want to come to and talk about today. This, like I like to think, everything we do in the lab eventually has some clinical relevance, and part of the reason for doing this is the truly unsatisfactory nature of our current classification, in that these traditional categories are often pretty loose with how they're defined. They're often distinguished only by a single marker, which is often difficult to profile, such as MPO or myeloid peroxidase, the myeloid marker. And as I mentioned, not by really biological or genomic features. And often it's just a single marker that can distinguish these entities that then become of such great clinical importance for how patients are treated as to whether they're classified as ETP-ALL and perhaps treated with a lymphoid regimen, or classified as mixed phenotype acute leukemia that might invoke a more myeloid regimen. There are additional reasons to be interested in this, and that's as we think about the current era of immunotherapy and CAR T-cell therapy in particular. Of course, CD19-directed CAR T-cell therapy has been such a great success story, but one mechanism of resistance is that these lineage-ambiguous leukemias, some of which are driven by MLL, for example, can undergo plasticity or lineage switch with change in their expression of cell surface markers, leading to lack of sensitivity to these agents. So there's a real rationale therapeutically as well to better understand these leukemias. So what is the cell of origin? And a nice review from Alejandro Gutierrez and Alex Kensis a few years ago was talking about where these lineage-ambiguous leukemias might arise. And if we think about the cases that have a T-cell feature, so T-myeloid leukemias, there are various theories that have been advanced. So for example, do the mutations arise in a very early stem cell that then differentiates into an ambiguous state? Do they arise in a later progenitor that retains the ability to express T and myeloid features? Or do they arise in a more truly committed T-cell progenitor, but then perhaps the mutations cause dedifferentiation and loss of specification of that T-cell lineage? And really, there was not a lot of direct data to support any of these three very reasonable hypotheses. So to show you these data today, again, let's step back in time a decade ago to our first foray into this, when this entity of early T-cell precursor leukemia was described. So it was considered to be a subset of T-ALL because it expressed a key marker of the T-cell lineage, CD3 in the cytoplasm, but it lacked expression of other key markers like CD1A, surface CD3 and CD8, and also had this striking pattern of aberrant expression of myeloid and stem cell markers. And because of a couple of features, the kind of immunophenotypic similarity to a mouse early stage of thymic maturation and gene expression features, it was termed early T-cell precursor leukemia because it was felt that leukemia was most similar to this ETP stage of early thymic T-cell development. But otherwise, everything was different. The mutations were very different from typical T-cell leukemia. Gene expression profile was very different from typical T-cell leukemia, as well as the poor outcome. And so, in fact, as the first genomic sequencing study by the Pediatric Cancer Genome Project way back in 2012, it's hard to believe it's a decade ago now, almost a decade ago, we picked ETP-ALL as being one of these least well understood high-risk forms of leukemia to try and understand drivers. And in fact, the initial results were a little frustrating. We didn't find a unifying fusion like some of the other things I've shown you before, or unifying sequence mutation. But as we delved deeper and sequenced more cases, what we found was two main observations, and the first of which was the mutational profile involved these three pathways that you can see are very myeloid-like. Myeloid transcription factors, often chromatin-modifying genes, are often seen in myeloid leukemias. And secondly, in the first of a number of great collaborations we've had with the John Dick Group in Toronto, we leveraged some of their human normal cell and leukemic cell transcriptomic data sets and showed that the gene expression profile of ETP-ALL actually looked the complete opposite of a normal human early T-cell precursor, and in contrast looked much more similar to an early hematopoietic stem and progenitor cell, suggesting that at least at the time of the leukemia being established, it looks most like a stem cell. The cell of origin, though, remained to be determined. Now, if we move forward a few years, if we come back to the TSNE and B-ALL I showed you before, a subtype I've not really spoken about is this form of leukemia that has recurrent rearrangements with another transcription factor, a zinc-fingered transcription factor called ZNF384. So these are all B-ALL cases. They're diagnosed as fulfilling the conventional pathologic criteria for B-ALL. But in parallel, we were a postdoc fellow, now faculty at UNC, Thomas Alexander, was leading a study examining mixed phenotype acute leukemia T myeloid or B myeloid leukemias, as shown here. And we found that the B myeloid cases, so expressing CD19 and myeloperoxidase, as shown here, the majority of them, about 2/3, had exactly the same rearrangements with ZNF384 as the B-ALL cases I showed you on the previous t-SNE plot for B-ALL. If we delve deeper into the genomics, we see that the co-lesions are basically identical, regardless of whether they're diagnosed as B-ALL or B-myeloid leukemia. And if we look at their transcriptomic outputs here, a subset of the t-SNE plot here, the ZNF384 rearranged cases occupy the same transcriptomic space, regardless of their immunophenotypic diagnosis. So to us, that suggested from the clinical perspective as a first step, this is a subtype that's defined by these rearrangements, and it transcends the primary immunophenotype. But now, what does it mean biologically? So what's the basis of this lineage ambiguity? And what's the basis of this feature that we often see where individual cases may have multiple immunophenotypically divergent subclones in a single case? So the answer here is this plasticity or variability is still intrinsic. It's not from mutational variegation. And we know this because we sequenced these subclones for many of these cases, and their co-lesions are basically the same. They don't acquire a notch or a FLT3 mutation or something and then diverge into a different lineage. In contrast, if you take primary leukemic cells and engraft them into a permissive mouse, but do it in a way in which you purify these subpopulations first and treat each one individually, remarkably, you see that the engrafted leukemias, even though you started with a subset of cells, the leukemias that come out at the back end are recapitulating this diversity. So the immunophenotypic plasticity is inherent to every leukemic cell. So this experiment suggested to us that this really is a seed and soil notion where you have the right initiating lesion in the right cell of origin, and it sets up this plasticity in the case. And we see this in other levels that I won't show today. We see it in patients as they evolve through disease. Their lineage can vary, as I mentioned before, for CAR T-cell therapy. And also we've seen it in a knock-in mouse model that I don't have time to present today, where we show that enforced expression of some of these fusions results in this skewing of hematopoietic development. Now, the last data I wanted to show today is a very recently published study that seeks to take this one step further. And so I think the work we've done before, I like to think is good, but it's been limited in one singular regard, and that is we've often started from a very confining immunophenotypic starting point. We've started with B-ALL, we started with T-ALL, we started with mixed phenotype leukemia, but we've clearly now seen that some of these entities don't respect those older criteria, and they can transcend these classification parameters. So what if we then step back and perform a similar analysis with a very large group of leukemias that really encompasses all subtypes of acute leukemia, B, myeloid, mixed phenotype, and T-ALL. A large collaborative study with multiple groups, particularly with the COG and the Munich Leukemia Laboratory, and then subject it to the standard analysis profiling approaches that I've shown you before. So this is a starting point. We see here many of the B-ALL subtypes that we've been talking about so far. But then we have this additional group of cases here that's much more myeloid mixed phenotype and T in flavor. Now, there were many very interesting sub-stories to this study. There are some additional subsets of leukemia that also were not previously recognized that I won't go into today. But I want to finish on this very striking story, and it's this subtype here, where about 63 of these cases were found to have a very distinct gene expression profile, all of which harbor deregulating structural variants involving the transcription factor BCL11B. So this is a subset of the T myeloid group. The cases here are colored by their primary diagnosis, so black for AML, green for T-ALL, and then purple for mixed phenotype and red for ETP. And so immediately you can see this is another entity that transcends subtype, where it comprises approximately a third of T myeloid mixed phenotype, a third of ETP-ALL cases. And for those that do like their immunophenotyping, it has a rather distinctive immunophenotype where it has variability for myeloperoxidase, which largely accounts for whether they're diagnosed as T myeloid or ETP, but otherwise a very strict phenotype with negativity for CD5 and other T cell markers and positivity for these myeloid and stem cell markers. We took some care to verify this discovery. We looked at a cohort of cases from from ECOG and showed exactly the same thing, that approximately a third of ththese T myeloid and ETP ALL cases belong to this same group. So why show this? Well, I think it makes very important insights into the cell of origin story that I started introducing in this section. So first, let's look at the genomics. So I mentioned that all of these cases have rearrangements of BCL11B, which is shown here. Each row here refers to one of the 8 different partners of rearrangement that are deregulating BCL11B, and each line here is the breakpoint spanning the BCL11B locus. They all spare BCL11B, preserving the coding domain. There are a couple of rare fusions, but they are a minority. So Lindsay Montefiore, a postdoc in my lab, led a lot of the downstream analysis and started by asking, why do we see this rather disparate group of fusion partners when obviously many genes could potentially be rearranged to BCL11B? Well, firstly, looking at what we know about BCL11B, it's a well-credentialed gene both in T cell development and also TALL, where it's known to be targeted by loss of function mutations and deletions in typical TALL, and also is rearranged as an enhancer donor to other oncogenes that define specific subtypes of TALL. It's not typically expressed in hematopoietic stem and progenitor cells, in contrast to normal T cells, and so this really led us to the hypothesis that this is an enhancer hijacking event where BCL11B may be aberrantly expressed in a hematopoietic stem cell. And so that is the case, and here's some of the evidence to support this. At a first level of analysis, Lindsay looked at CD34 cord blood-derived profiling based on K27 acetylation and showed a pattern such as this, where these are the breakpoints at one of the partners, ARID1B, and they're clustered around this super enhancer that's present in hematopoietic stem and progenitor cells. This is true for all of the partners of rearrangement. They're all documented super enhancers in HSCs. Another example is shown here. This is another cluster of rearrangements in the MYC downstream super enhancer region that's rearranged in a number of different neoplasms, but here again we see this cluster of breakpoints at the BENK enhancer, the B cell enhancer, in a subset of these leukemias. Lindsay then went on to demonstrate that these enhancers do indeed deregulate BCL11B using a high chip approach that incorporates K27 acetyl profiling with chromatin topology analysis. So these are normal CD34 cells where we see no interaction between one of the partners, again ARID1B, and BCL11B that here is inactive in CD34 cells. But in contrast, if we look at one of the cases that has a rearrangement, we see activation of BCL11B by gene expression and acetylation, and evidence of looping between these two loci between the proximal ARID1B region and BCL11B. That was seen with the conventional targets of rearrangement uniformly. There's one additional story here that was very interesting, that a fifth of these cases that deregulate BCL11B had an entirely novel mechanism of neo-enhancer generation. So rather than a translocation, they exhibited this tiny little 2 kilobase region that was amplified almost a megabase downstream of BCL11B to what appears to be the real sweet spot level of genomic amplification, about 15 to 20 copies of the gene, in contrast to a sample here that does not have this amplification, that we could verify by genomic PCR and show that it was associated with enhancer RNA expression in these cases and only these cases. And so what is this doing? Well, there's very low level acetylation of this region in normal cells as shown here, but in the context of this application, there is basically establishment of a fully fledged or credentialed enhancer region that then activates BCL11B. We see this if we look at these cases that have this amplification, extremely high level of K27 acetylation that we don't see in other cases in this group or other TALL cell lines. And if we use the same high chip approach again, we see that this amplified enhancer is in fact generating quite a large TAD, if you like, that isn't just this enhancer, but also co-opting another distal enhancer to create a large TAD that's then deregulating BCL11B. So is this originating in a hematopoietic stem and progenitor cell? And I'll just finish with a couple of observations that makes this point. In another collaboration with the John Dick group, we took all of our transcriptomic data from our cohort of leukemias, and onto these we mapped the transcriptomic signatures of stem and progenitor cells. These are the BCL11B cases shown green here, and we see that they're enriched for the transcriptomic signature, again, of stem and multipotent progenitor cells. A complementary approach is to look at individual cases by combined single-cell ATAC and gene expression profiling. And again, we see this very strong enrichment in these leukemias for these activated and long-term hematopoietic stem cell signatures. Now, again, we might be criticized that that is somewhat correlative and observational, but not directly demonstrative. One other observation that I think is very telling is that Shunsuke Kimura, another postdoc in my lab, is studying TAL, look very closely at the patterns of T-cell receptor gene rearrangement in these leukemias and showed that not a single one of these cases showed any evidence of rearrangement of any of the T-cell receptor genes, in contrast to other ETPs and MPALs or conventional TALs, very strongly suggesting that the cell of origin is prior to any initiation of a normal T-cell development program, so probably a pre-thymic progenitor cell. And finally, we could show that these are indeed transforming both in mouse and human if we introduce BCL11B and FLT3, with which mutations which are common in this group, we see colony replating. And if we do the same experiment in cord blood cells, we see emergence of a population of cells that's now lineage aberrant and expressing cytoplasmic CD3. So there I'll stop. And to try and bring this back to 5 very simple conclusions that I think are important. They're influencing how we continue to work in the lab at St. Jude and with our collaborators, and how we think about diagnosis and treatment. Genomic analysis continues to refine the constellations of drivers that underlie acute leukemia. This is still a work in progress. Genomic characterization is clinically important, and contemporary diagnostics require sequencing-based approaches. Genomic subtype may transcend traditional pathologic criteria, and so I think we'll continue to see evolution in the classification of leukemia along these lines. And as I've shown you, genomic characterization can identify often unexpected therapeutic targets. And finally, I've given you an example where perhaps protein degradation can be useful, but perhaps even more important is that most of the cases of childhood ALL are driven by transcription factor deregulation that's historically considered undruggable, and clearly targeting perhaps by protein degradation represents an exciting new approach to try and accomplish this goal. There I'll stop and thank my wonderful group. I just want to single, really everyone in the group has contributed at various levels to these studies, but I particularly want to thank Catherine Roberts, that drove many of the studies of PH-like leukemia, Xiaohui Gu, now faculty at City of Hope, together with Lindsay Montefiore, that did much of the work in Lynch ambiguous leukemias. I mentioned the group in CBT for the protein degradation project. Many collaborators at St. Jude, particularly Jingwei Zhang and her group, Sam Brady, Jeff Clow and his group for the mixed phenotype studies, and our collaborators at COG, ECOG, as well as the Munich Leukemia Laboratory. And thank you all for your attention.
Fantastic talk, Charles. Thank you for kicking off the series in such a strong way. Dr. Mulligan has agreed to stay for questions for some time, so we will move into them. And for those of you that need to leave at the top of the hour, we're back next week, and we'll continue on at this time for the next 15 weeks. So starting out with the first question, Keito Hoshizuki asks, how similar or different are the genetics of infant ALL compared to that of childhood ALL?
That's a great question. So I think a couple of observations have been made. So of course, infant leukemia is very much enriched for rearrangements of MLL. So firstly, there's a difference in that MLL rearrangements are much more common than in later ages in childhood. Secondly, MLL really is another example of a sufficient oncogene, and so those cases have the lowest burden of secondary mutations of any form of leukemia. Now that does differ from MLL rearranged leukemia later in life, both in children and adults, where there is an increased burden of additional alterations. The second observation is that there are other subtypes of leukemia that are more common in infants, and one example of those is NUTM1 rearranged leukemia, which I've not spoken about today, but which is very interesting because NUTM1 is also a target of rearrangement, particularly to BRD4 in midline carcinoma, but in infant leukemia in particular, it's rearranged to another number of other targets, BRD9 as well as others. So they're probably two of the more striking observations of differences in infancy and leukemia from the genomic perspective.
Great. T. Oliver asks, why do you think your substitution signature extraction method is finding UV-related mutagenesis? Can you explain how UV might be involved? Do you see it associated with the indel signature? And if not, wondering whether there's, in fact, a novel signature that's most closely relates to SBS7. So the genesis of that signature.
Yeah, it's great. I mean, you know, we spent a lot of time thinking about this. Sam and Jing Wei and her group have done, you know, a great deal of analyses of these signatures in leukemias and other tumors, but they do not feel as a novel signature. It is the UV-induced signature, and it suggests that that is the mechanism of mutagenesis. You think, well, are children really exposed to UV light and that's generating mutations? Well, it's possible, but the answer is, at least based on the genomic profiling and analysis of those substitution patterns, this appears to be the UV signature. Why it predominates in these groups, those three aneuploid groups, we don't know, but it kind of it is tantalizing to suggest that you have this, you know, Big Bang aneuploidy prior to birth or soon after birth, and then this second pathway is active, at least in a subset of cases.
Excellent. This next question, I think, reflects the impact and importance of the global audience. So Pratik Bhatia asks, what are the most important newer genetic BAL entities that you would suggest to be incorporated into a diagnostic workup, excluding Philadelphia-like? And what diagnostic algorithm would you suggest for a resource-constrained laboratory where whole exome and RNA-seq can't be done in all cases?
Yeah, this is a really important question, and it's an area that we're very actively involved with, working with a number of our partner sites, you know, with St. Jude Global around the world, and a number of groups are doing this. Thomas Alexander, I mentioned, is pursuing this with other approaches. So I think, yeah, we're only just starting to understand the interaction between these newer subtypes and prognosis. And I had to hurry through the data, but it's interesting that in children, we're seeing strong association, even in the context of MRD risk-adapted therapy for some of these new subtypes. So some of them are associated with increased risks, such as ETV6 RUNX1-like, some of which are not neatly diagnosed by a single approach. In adults, if that's of interest to you as well, some of the associations appear a little different, although we're still lacking MRD risk-adapted trial data to look at those associations. That will come in a little while. So I think it would be wrong to say there's only a couple of additional subtypes, unfortunately, for your question, that should be done in addition to PH-like leukemia. And unfortunately, some of the new subtypes of interest, they may not have necessarily an immediately applicable target to go after, but they are of interest for prognosis, because often, you know, diagnosticians or clinicians come back and say, we have high MRD or a failure of therapy, what's the form of leukemia? So I would strongly advocate that there should be an agnostic approach at some level to diagnose leukemia. And this is one of the approaches we're taking. Can we implement RNA-seq-based diagnostics, because it captures as a single modality pretty much everything you need, or an alternative, you know, cost, a more effective approach such as nanopore-based sequencing, and that's looking very promising. Now, if that's simply not possible, there are a couple of focused approaches. For example, some of the new good risk subtypes like DUX4 rearranged leukemia has a particular marker that can be used in flow. The same for PH-like leukemia for serial F2. And there are a few other nuances like that that can be built into more conventional pipelines. But, you know, for better or worse, really to do the best by our patients in all settings, particularly as we don't fully understand how those prevalences of subtypes vary in different regions around the world, some level of sequencing-based approach is desirable.
Excellent. Alex Gout asks, the Philadelphia-like grouping appears to involve a variety of different driver events and fusions. Do you feel that further study of this group is going to render more refined subtypes that potentially represent each of these specific driver fusions? Or do you think keeping them grouped as a more broad PH-like category will be beneficial?
Yes, it's a good question, Alex. I think it is important, both from understanding the biology, but also for classification. And we're seeing this already. You know, we've gone through a few iterations, for example, of the RNA-seq pipeline, and we've seen some of these entities like PH-like, but also the PAX5 alt group that I didn't really talk about. The more cases you have profiled the same way, the more it kind of crystallizes or nucleates, if you like, and you see subgroups starting to emerge where you think, aha, well, now the biology seems more logical. Now, PH-like is just more diverse. The drivers are very diverse. Their co-lesions vary a lot. I don't think we'll ever see, true Christmas of that one group. There is some bleed over to other groups, but in part that's expected because sometimes the same co-lesions are shared. Aneuploidy, for example, or an ECROS mutation or a PAX5 alteration. But yes, I believe we've still got a way to go to fully understand the grouping of these leukemias.
Great. The next question was, what is the nature of these mutations? Are they single base pair, indels, and are they occurring randomly throughout the genome or at predominant specific regions of the DNA, like repeat regions, et cetera? Is a question from Vibhor Mishra.
Okay, it's a good question. We have looked at that as well. I mean, one of the readouts that's been used for regionality is contagious, that phenomenon that's been described by the Cambridge group, and we don't really see that. I think we've seen a couple of examples where there's clustering of mutations at specific genes. The mutations, there are SNVs are more common in indels, certainly in ALL, and non-silent variations are more commonly SNVs than indels. The indels that we do see, they do predominantly affect key genes, such as things like FLT3, IL-7 receptor. There are a number of interesting genes, particularly in the cytokine receptor signaling pathway, where indels predominate. But overall, SNVs are more common than indels.
Question: so you showed the mutation burden is very low in these cancers. What does this mean about the mechanism of how these form? And what does it mean? You've highlighted some important subgroups in kinases. What does the simple genome mean for the therapeutic approaches to other types of leukemias that are not easily tractable with small molecules?
Okay, very good questions, very different questions. So I guess one kind of area I didn't go into, which is somewhat more speculative, but a lot of people have been thinking about, more philosophical perhaps, is, what's driving the initial acquisition of the mutations and the fusions? And obviously we know something about the germline predisposition. There may still be a stochastic element or something subtle environmental that's allowing a topo generated rearrangement or something that really primes the system. One thing that we do see, I mentioned early on, is that the burden of copy number alterations is higher than variants, and that's really important. Many of the key alterations that we recognize as driving deregulation of pathways, B lymphoid development is a good one, but it's only one, is driven by focal deletions. And we know that many of those deletions are driven by, or at least have the footprint of, the recombinase activating gene machinery that normally mediates antigen receptor gene rearrangement. We also know that there is a peak in onset, at least of the commonest types of ALL, at that kind of infection exposure time when kids are typically perhaps weaning off maternal immunity and evolving their own cellular immune response very quickly. So to my mind, at least, this kind of brings together two notions of, this theory that's been promulgated for some time by Mel Greaves and Kinlan and others about, the delayed mixing or infection hypothesis that, kids that are exposed to a battery of infectious agents at about that time point are prone to developing leukemia. But it also is now bringing that together with the genomics. So to my mind, as I conceptualize it, I think that there may be a priming event in utero or perhaps early in life. And then when this lymphoid compartment is undergoing brisk expansion, perhaps at a time when the child is exposed to pathogens, that RAG machinery is really, really active. It's driving generation of that antibody maturation response, but it's also picking off some of these other genes. And when they confer a selective advantage, they're additional important steps on the road to leukemia development. That's harder to test, of course, but there is some evidence. So not from my group, but from others that have used infection exposure type mouse modeling. They've shown that pathogen-free mice show a very retarded development of ALL compared to those that are exposed to pathogens. So I think now there is some complementary genetic and experimental data that supports that notion. Implications for other leukemias. I mean, that's a really broad topic. I mean, I think it's, in some ways, the sparse genome of ALL is to our advantage for some of the reasons I've shown today. And we know that many of the drivers are clonal, and despite the mutational evolution, those clonal drivers are still there. That's probably true for other leukemias as well. But if one looks at adult myeloid leukemia, for example, the drivers are very different. So obviously very different approaches are being developed there, targeting, epigenetic programming, IDH mutations, etc. So I think part of what the strategy may mean or may be is still going to be very much dependent on what we consider to be the driving clonal lesions.
Great. Feel free to continue to type in questions for Dr. Mulligan. Next question. You showed in some cases that there are non-coding SNVs or mutations. How common is this? Are we at the tip of the iceberg and this is going to be incredibly prevalent across leukemias or other cancers, or is this a rare event?
Well, I mean, the simple answer is that non-coding variations are really common. I mean, if you look at the number of coding versus non-coding variations, the non-coding ones outnumber them, but in part that's because the non-coding space is larger. The key question is how many of them are drivers, and there are some lessons that have been learned already. So, for example, if one looks at non-coding structural variations, they're often really important. So, for example, some of the structural variations that enhance a hijacking or indeed result in chimeric fusions break in non-coding regions. And that's why some of these alterations are not identified by other approaches and may not indeed be identified by RNA-seq. So the BCL11B example is a good example of that. That's perhaps more true for TALL than BALL, where we and others have shown in our previous studies that non-whole genome approaches have a lower yield than in, say, in BALL, where many more of the cases are rescued by RNA sequencing. A second part may be sequence mutational drivers. So again, TALL has been very telling. There's landmark papers from Tom Look and Rick Young showing that some of the subtypes of TALL that for some patients are driven by fusions and rearrangements, in others deregulate the same oncogene by an enhancer mutation or an enhancer creation mutation. So in some ways a little bit similar to BCL11B. Now, what have we got to learn? I think we've got a lot to learn. And I think part of the reason why we are still learning is that we've just by necessity in how genome sequencing has progressed, we had to bite off what was tractable. And so a lot of what we've been learning has been from RNA sequencing. Now whole genome sequencing is kind of playing catch up. And I think for us, it's going to be a really exciting time in the next few years where we do have much more comprehensive integrated genome and transcriptome sequencing, so we can more comprehensively make these associations that you're asking between non-coding variants and their effect on oncogenes. But we do know that from looking at cases that are non-classified, we're just not rescuing a good proportion of them still. So I suspect we're going to find some exciting stuff to come.
Great. Wendy Stock, actually amongst many of the others, thanks you for the beautiful talk. But as a follow-up, do you have evidence or suspect the Big Bang theory for adult for aneuploidy in adult leukemias, or is this something that's unique to pediatric cancer? Do you have data there?
It's a great question, Wendy. I mean, the beauty of what we did, I think, was because we had a nice data set to play with. That is, we had aneuploidy with chromosomal gain, plus we had obviously we had the data, we had the whole genome sequencing data, and we have less whole genome sequencing data to allow that kind of broad SNV math profiling in adult leukemia. The other challenge is that you don't see hyperdiploid ALL so much in adults. We do see it, in some patients, for example, in pH positive leukemia, but much less common, and we don't have enough data there yet. I would love to be able to answer this for hypodiploid leukemia for a couple of reasons. It's more common in adults, and also we know that some of the hypodiploid cases look really similar to hyperdiploid. So haploid cases, although they very dramatic differing patterns of aneuploidy, many of the co-lesions are the same, the transcriptomic profile is almost identical. So I do think there are some similarities in genesis between at least some of the hypodiploid leukemias and hyperdiploid. Harder to answer though, because there we're dealing with chromosomal loss and we can't do the same analysis. And I haven't thought of a way yet to do it. Perhaps someone can tell me. So I think that's a great question, Wendy, but to be answered.
Farah Nassar asks, have there been epigenetic studies that would explain the BALL subtypes or would explain the effect on patient prognosis? So what can you learn from the epigenetic characterizations?
Yeah, this is a really, really good question. And so many groups have been doing this. We have work that's in revision at the moment, looking at genome-wide whole genome bisulfite sequencing and ALL. And I think, there are some lessons to be learned. And I think it's also interesting. One of the great things about being here at St. Jude is we're talking with Charlie, with my colleague Paul Northcott in the brain tumor space and others, and looking at what methylation, methylome and other epigenetic marks look like across pediatric cancer. So, for example, I think, methylation has been incredible in brain tumors where it seems that even better than RNA sequencing expression, it crisply defines out many of the newer subgroups of brain tumors. When we look at ALL, despite our best analysis, yes, we can find methylation signatures, but they're just not as distinct as what we see from gene expression. So what does that mean? I mean, I think in part it may reflect that we may be dealing with, at least for most B-ALL cases, and maybe a lot of T-ALL cases, a somewhat more narrow cell of origin range. We're dealing with that kind of pro to pre-B cell stage, for example, whereas perhaps in the brain tumor space, there are many more diverse, variegated cells of origin that have a much more distinct methylation footprint on the final tumors. But we do see differences. We see particularly the methylome has a stronger signature in the subtypes that are driven by key transcription factor rearrangements than, for example, pH-like leukemia. A subset, but only a small subset of those differentially methylated regions are associated with gene expression. So it appears that a lot of that transcriptomic output is determined more by the primary genomic alteration than an associated or secondary change in methylome. And in terms of outcome, there's some association. I think the data are better for T than for B, and the associations with outcome tend to be broader rather than refining subtype. There's this CIMP methylation phenotype that's been reported. So I think there's probably still information to be milked there. Again, I think perhaps we may learn more as we get more integrated data between whole genome platforms, but it doesn't seem as striking to my mind, at least, as some of the other pediatric tumor contexts.
Great. Xuan Zhang asks, you demonstrated the recurrent mutations observed in ETP ALL, highlighting the co-occurrence of RAS signaling mutations and alterations in PRC2, which is also observed in other cancers. Do you think the co-occurrence of mutations in RAS and PRC2 is largely by chance, or that they rely on each other for promoting the tumorigenesis?
I'd like to think nothing in cancer is by chance. That's a personal viewpoint. Well, I mean, I think they clearly co-occur. So what drives their original origin, that may be chance or something else, but they're clearly selected for. So they clearly have a pathogenic role. And there's a large number of models now, not so much from my group, that have picked off many of these lesions and programmed them in alone or together into mouse models and have shown that at some level, whether you activate the IL-7 receptor signaling pathway, that JAK-STAT pathway, the RAS pathway, or indeed you perturb epigenomic modulators or regulators, you can initiate leukemias that at least at some level recapitulate ETP ALL. So I don't think their co-occurrence is random by any means. I think there's a reason for that biologically.
David Wheeler asks, in the ETV6-like class of leukemias, is it possible that an ETV6 fusion did exist at an earlier time in development of the disease, but was subsequently lost?
Yes, possibly, Dave, but I think there still has to be a driver that's sustaining the leukemic clone. And I think that's what we've seen in most cases. So, for example, again, I would sped through it, but there's a nice study from June Yang and Kim Nichols when they've been looking at ETV6 germline mutations, that they show that many of those cases of childhood ALL that are driven by a germline ETV6 mutation have that mutation and then a second deletion of ETV6. And that's also seen for ETV6 RUNX1 leukemia. You have the fusion and then a second copy deletion. Plus many of the other cases that don't have the germline ETV6 have two lesions of ETV6 if they're not one of the cases that has an alternate fusion. But you're still very probably correct in some cases, because we know there are many more children that are born with ETV6 RUNX1 leukemia that eventually develop the disease. But to me, it's not persuasive that initiating event then disappears and nothing else is needed as a surrogate. I think from what we've seen profiling those cases, there's usually some other lesion or multiple lesions targeting the ETV6 machinery or complex.
Great. Two somewhat technical questions. One from Dikshit Gopal Gupta, who says, what is your opinion of the use of ruxolitinib with dasatinib? It decreased the phospho-STAT5 at the initial time points and returned to baseline by 72 hours of continued drug treatment. study from Sarah Tazian's group published in JCI in 2021. Can we employ a quantitative eye-track mass spectrometry technology at baseline and at 24, 48, 72 hours to target the deregulated proteins at different times and inhibit by PROTACs to elucidate newer potential therapeutic targets?
Wow, that's a great question. Yes. So I think, yes, I mean, Sarah's done some fabulous work there, teasing out mechanistically how these pathways cooperate. And there are a number of studies from various groups that, again, speak to this point that it's not just a single signaling pathway, that it's multiple. And in data I've not shown yet, we've shown, I guess, in an analogous way to what Sarah has shown, that if we take a key PROTAC, and we particularly use a PROTAC that is incompletely effective, and we then do a screen with a panel of kinase inhibition, one can get really spectacular suppression or eradication of the leukemic cell. So SARC or ABL inhibitors like dasatinib are a standout, but there are several other key targets as well that show that very strong synergy. And yes, I agree with you. I think I haven't seen enough, I haven't generated enough data yet to show what's going to be the best biomarker. That's been a struggle, and how well that could be implemented. But that's a really nice notion that one could use that approach early on and then use it as a correlative marker to then look at responses in patients. I also think that one thing that's really important in that field is that I think a lot of us have been discouraged because we've asked for the biggest result in the hardest context, and that is to give a partially active agent like ruxolitinib as monotherapy in heavily pretreated disease. And we know that the first bite of the cherry is the best, and the best chance for success with these newer approaches is early. And I think that is where a lot of the field is moving. If it's later in therapy at relapse, and often that's how trials need to be done, of course, then it's not now a single agent, particularly one like this that we know is more variable, but it's combined. Or for our studies here and others, we progressively move the logical inhibitor up front to give it the best chance of success.
Great. A question that I missed from Sarah Sherman. Sorry, Sarah. How do you think single cell technologies will impact the landscape of known leukemia subtypes?
Yeah, well, that's a really good question, and there are multiple ways to do this. I mean, I think so will the deployment of single cell technologies change subtyping? I don't know, because it's still, to my mind, I think the subtypes that we've recognized well appear to have a clonal driver. There are lesions that wax and wane through evolution, of course, but particularly in childhood ALL, there are exceptions, of course. You see biclonal leukemias, and there are always exceptions to every rule. The clonal drivers are typically present in all cells. So there, conceptually, single cell technologies won't offer an advantage in most cases in the classification, but they're clearly really fantastic for a variety of different things. So whether it's understanding influences on subsets of leukemic cells, understanding the timing of origin, where one can couple more complex readouts, site-seq, as well as mutational fusion profiling. One can understand clonal heterogeneity with much greater precision using DNA single cell approaches like the tapestry, for example. So I think they're going to continue to be highly impactful for many reasons, but probably less so for overall subtyping, but perhaps I'll be proven wrong.
Great. Technical question from Pratik Bhatia. How do we define driver mutations in terms of clonal variant allele frequency? I mean, from the number of n number of somatic mutations in next generation sequencing, which ones would be termed driver and which passenger and which cooperative if we wish to integrate fusional, ploidy, and somatic SNV indel data?
Right. I mean, great questions. And I guess there are various ways to answer that question, and many of them are in silico, and some of them, of course, are experimental. So I didn't show any of these data, and some really, amazing work done by Sam Brady and Jing Wei's group looking at this huge cohort who identified many new drivers, new pathways that are not previously recognized, and also constellations, co-associations of lesions within subgroups or across subgroups, and also patterns of exclusivity. So I guess at the simplest level, we've called driver from a computational statistical perspective, deploying tools that account for multiple types of lesion, tools such as GRIN or Mut6CV, etc. So purely computational. I think also using a more bespoke approach where, again, you look at the data and you say, wow, all these things are always co-occurring, or they're always mutually exclusive, or they're only in this group. Those sorts of observations may be less statistically rigorous, but then I think making compelling suggestions that the lesions are drivers. Ultimately, to my mind at least, I think the proof of the pudding for a driver is its experimental confirmation, that it's introduced or manipulated or its dependency tested and it's shown to have a biological effect. But that's tough because now we know from, the more you sequence, the more mutations you find, the more genes are altered and the more putative drivers there are. But they're kind of the approaches that we've used to document or think about what defines a driver in ALL.
Great. So Dr. Mulligan agreed to stay for a half hour. I think we're there. So I want to thank him for a phenomenal talk. Next week is Dr. Meredith Irwin from the Hospital for Sick Children, where she's pediatrician in Treat. We'll be talking about identification of biomarkers, models, and targets to improve the outcomes and precision approaches for neuroblastoma. The week after that is Dr. Olivier Delattre coming to us live from Paris, where he will be talking about modulation of the activity of the dominant oncogene EWS-FLI1 by germline polymorphisms and cohesion mutations. And so, Charles, again, thank you for the phenomenal kickoff to the series, and we look forward to seeing everybody next week.
Thanks, Charlie, and thanks, everyone. Thank you for joining.